Okay, so let's start with cytoplasmic organelles. Inside the cell membrane, the fluid cytoplasm or cytosol bathes metabolically active structures called organelles. So organelles are in the cytoplasm and they are metabolically active, which may be membranous such as mitochondria or non-membranous protein complexes such as ribosomes or proteosomes. Most organelles are positioned in the cytoplasm by movement along the polymers of cytoskeleton, which also determines the cell shape and motility. So we have organelles in the cytoplasm, membranous like mitochondria or non-membranous like ribosomes and proteosomes, and they are held in a place in cytoplasm through the help of cytoskeleton. Cytoskeleton is also used for the shape of the cell and the mobility. Cytosol also contains hundreds of enzymes, such as those of glycolytic pathways, which produce building blocks for the larger molecules and break down small molecules to liberate oxygen. So, glycolytic pathway is basically is a pathway that produces oxygen, that produces energy, and we will study glycolytic pathway in detail in the coming chapters. We have all studied glycolytic pathways in our pre-med, but still, oxygen, carbon dioxide, electrolytic ions, low molecular weight substrates, metabolites, and waste products all diffuse through cytoplasm, either freely or bound to a protein, entering or leaving organelles where they are used or produced. So these are the various roles that an organelles play sometimes, you know, producing oxygen, carbon dioxide, metabolic waste, etc. And these things either are present freely in the cytoplasm or associated or bound to a protein. So ribosome is the first organelle we're going to study. Let's see how dynamic the ribosome is. Ribosome are macromolecular machines. So they are responsible for the producing the polypeptide chain, the translation. About 20 to 30 nanometers in size, which assemble polypeptide from amino acid on molecules of transfer RNA in sequence specified by mRNA. So there is an mRNA, and what ribosome does is, with the help of tRNA, brings the specific amino acid and associates it with its mRNA, eventually forming a polypeptide chain. A functional ribosome has two subunits of different sizes bound to a strand of mRNA. The core of the small ribosomal subunit is highly folded ribosomal RNA, that is rRNA. rRNA is the RNA produced by ribosome chain associated with more than 30 unique proteins. The core of the large subunit has three other rRNA molecules and nearly 50 other basic proteins. So there are two subunits of the ribosome. One subunit is smaller, that is associated with more than 30 unique proteins, and the other associated with 50 basic other proteins. The rRNA molecule in the ribosomal subunit not only provides structural support but also position transfer RNAs molecule bearing amino acid in the correct reading frame. So the role of rRNA is to not only just have a structural support of the RNA, of the ribosome, but its important role is to position the tRNA on the mRNA, and tRNA is the one RNA which is consisting the amino acid. It brings the amino acid from the cytoplasm. These ribosomal proteins are themselves synthesized in cytoplasmic ribosomes. Okay, but are then imported to the nucleus where they associated with newly synthesized rRNA. So ribosomal proteins are themselves synthesized in the cytoplasmic ribosome, but these proteins sometimes travel to the nucleus, and where they get associated with newly synthesized rRNA. The ribosomal subunit thus formed then move from the nucleus to the cytoplasm where they are reused many times. So this ribosomal subunit again comes back to the nucleus, from the nucleus to the cytoplasm, and then this ribosome can be used as an organelle in the cell multiple times. for translation of the mRNA strands. During protein synthesis, many ribosomes typically bind the same strand of the mRNA to form large complexes called polyribosomes. So, poly, always remember when we have a word poly means many and ribosome. So, polysomes are just the multiple ribosomes that help to that attach on the same mRNA, a single mRNA to produce protein, the polypeptide chain. In stained preparation, polyribosomes stain intensely basic. Polyribosomes are intensely basic because of the numerous phosphate group of the constituent RNA molecule that act as a polyanion. So because of the phosphate group, they stain basic. They are anionic in nature and they adapt the basic stain. Basic stain like hematoxylin. Thus, cytoplasmic regions that stain intensely with hematoxylin and basic dyes such as methylene and toluidine blue indicate sites of active protein synthesis. So when a cell is stained, where we see a patches of hematoxylin stain is around the ribosome, is where we know that we have a translation process going on. Proper folding of the new protein is guided by protein chaperones. Denatured proteins or those that cannot be refolded properly are conjugated to the protein ubiquitin that targets them for breakdown by proteosomes discussed below. So basically, proteosomes are important whenever a polypeptide chain is produced but is unable to refold itself. It's going to be unstable in the cytoplasm. That's why it's important that we cut it down and then again reframe it. We'll study how this works in detail. As indicated in figure 2.9, let's check figure 2.9. This is figure 2.9, polyribosome free or bound to the endoplasmic reticulum. So as we studied, these are the first small half of the ribosome. This is the larger ribosome. This pink here is an mRNA strand. This blue here is the misfold is the protein that is being produced, and the misfolded protein here gets denatured, and we'll see how it works. And let's go up again and check what happens. Protein synthesized for use within the cytosol, within the cytoplasm, like glycolytic enzyme, for example, or for import into the nucleus, and certain other organelles are synthesized on the polyribosomes existing as isolated cytoplasmic clusters. Okay. Polyribosomes attached to the membrane of the endoplasmic reticulum translate mRNA coding for membrane protein of the ER. So this is the role of ER. We'll study what how ER works, Golgi pair. Next is what ER. Let's check on what's happening with endoplasmic reticulum. The cytoplasm of most cells contain a convoluted membranous network called the endoplasmic reticulum. As shown in the figure 2.10, this network, the reticulum, when the word reticulum means a network, extends from the surface of the nucleus throughout most of the cytoplasm and encloses a series of intercommunicating channels called cisternae. With a membrane surface up to 30 times that of the plasma membrane, the endoplasmic reticulum is a major site for vital cellular activities, including biosynthesis of protein and lipids. Numerous polyribosomes attached. So the cisternae, the channels, these are important structural unit, the structural function of the endoplasmic reticulum. Cisternae basically means the reservoirs. So basically it's a structure that extends from the nucleus. It's in the form of a reticulum that is a network, and it has intercommunicating channels called cisternae. We can just highlight that what are cisternae. They are intercommunicating channels with a membrane surface up to 30 times that of the plasma membrane. So membrane surface area of the endoplasmic reticulum is more than what's of a plasma membrane. The ER is a major site for vital cellular activities. What cellular activities? Let's check. Including biosynthesis of proteins and lipid. So synthesis of proteins and lipid is what happens at the surface of the endoplasmic reticulum. Numerous polyribosomes attached to the membrane in some regions of the ER also allow two types of ER to be distinguished. So presence of polyribosome makes the surface rough. That's why it's called rough endoplasmic reticulum. And where we don't have polyribosomes, that endoplasmic reticulum is called the soft endoplasmic reticulum. Let's we'll refer it as RER. Rough endoplasmic reticulum RER is prominent in cells specialized for protein secretion. So where we have the ribosome, there we'll have the protein synthesis, such as pancreatic. Here are examples where the rough endoplasmic reticulum is dynamic and produces proteins. uh, Pancreatic acinar cells, fibroblasts produce collagen, plasma cells produce immunoglobulins, that is antibodies. The rough endoplasmic reticulum consists of sac-like as well as a parallel sacs of flattened cisternes, each limited to membranes that are continuous with the outer membrane of the nuclear envelope. So RER continues like a sac-like structure and it extends from the nuclear envelope, from the outer surface of the nuclear envelope. The presence of polyribosomes on the systolic surface of the rough endoplasmic reticulum confers basophilic staining properties to this organelle when viewed with the light microscope. So we know that ribosomes stain basic, and because ribosomes stain basic, the rough endoplasmic reticulum also appears uh, with the hematoxylin stain and its basic nature. The major function of the RER is the production of membrane-associated proteins, proteins of many membranous organelles, and proteins to be secreted by exocytosis. So the major function here needs to be highlighted and needs to be remembered that it produces mainly the proteins that are associated with the membranous organelles, like we have mitochondria, and also it produces the proteins that are exocytosis from the cell. Production here includes the initial core, glycosylation of glycoprotein, certain other post-translational modification of newly formed polypeptides, and the assembly of the multi-chain proteins. So this is also important function again, that after the the glycosylation of the glycoprotein, and like glycosylation, other translational modification also are are done by the endoplasmic reticulum. These activities are mediated by resident enzyme of the rough endoplasmic reticulum and by protein complexes that act as a chaperones guiding the folding of nascent protein, inhibition aggregation, inhibiting aggregation, and generally monitoring protein quality with the ER. So the most important word here is chaperones. These chaperones are enzymes that are present in the rough endoplasmic reticulum, and they are responsible for various functions like folding of the nascent protein, inhibition of aggregation, and generally monitoring the protein quality in the ER. So the role of chaperone here is very important and looks like a question to me. Uh, okay, so protein synthesis. Protein synthesis begins on polyribosomes in cytosol, that is in cytoplasm. The 5' strand of the mRNA for protein destined to be segregated in the ER encoded an N-terminal signal sequence of 15 to 40 amino acids that include a series of 6 or more hydrophobic residues. So mRNA has a 5 dash end and a 3 dash end. So 5 dash end is from where the nuclear, the N terminal, that is a signal sequence. What is a signal sequence? This is a sequence from where the polypeptide production, the translation will start. This N-terminal signal sequence is made up of 15 to 40 amino acids. As shown in the figure 2.11, we'll check figure 2.11. The newly translated signal sequence is bound by a protein complex called signal recognition particle, SRP, which inhibits further polypeptide elongation. The SRP ribosome nascent peptide complex binds to SRP receptor on the rough endoplasmic reticulum. SRP then releases the signal sequence, allowing translation to continue with the nascent polypeptide chain transferred to a translocator complex, also called transcolon, through rough endoplasmic reticulum. Figure 2.11. Here, figure 2.11 is the most important. We have a 5' end of the mRNA. This pink color here is an mRNA, and 3' end of this mRNA. This is a ribosome. We have a tRNA here, and we have a new polypeptide chain being produced. What happens here now is, in this being the rough endoplasmic reticulum, its role major function is to monitor the polypeptide production, and this blue color here is a signal recognition particle which gets attached here to the polyribosome, through which the polypeptide chain is being monitored. And this R red color here is the SRP receptor molecule. So now the receptor and the SRP molecule will attach, and this is how the interaction between the ER and the polyribosomes takes place, forming a ribosome receptor and protein translocator complex. So as the word suggests, we'll go by word, is ribosome receptor. Why ribosome receptor? Because it recepts the ribosome, it attaches the ribosome there. And protein translocator. Translocation is the process where protein is produced, and protein translocator complex. So this is a complex through which the polypeptide chain will produce. Also, one more molecule here is the signal peptidase. And we can see here a growing polypeptide chain which is modified and which is completed. The quality check has been done. This also, we have to remember that this translocator complex is also called transcolon. So whenever we have a word transcolon, we know that we're talking about this translocator complex. It is made up of the ribosomal receptor and the protein translocator. In the lumen of the rough endoplasmic reticulum, this is the lumen of the rough endoplasmic reticulum, the signal sequence is removed by an enzyme, signal peptidase. So this role of signal peptidase here is important. The signal sequence is removed because signal sequence was just to start a signaling sequence. It was just, its role was to start the polypeptide production, and it's removed later because it's not required in the final protein. So this type of small changes are done by rough endoplasmic reticulum, like the finishing of the polypeptide. With the ribosome docked in the rough endoplasmic reticulum surface, translation continues with the growing polypeptide pushing itself, while chaperone and other proteins serve to pull the nascent polypeptide through the translocator complex. So we also have the chaperones here that are important to pull the nascent polypeptide here down to the cytoplasm and to aggregate, not to aggregate, but to separate it from the mRNA. It's work of the chaperones. Okay. Upon release from the ribosome, post-translational modifications and proper folding of the polypeptide continues. So this is what has been explained here in this paragraph. Basically, Proper folding of the polypeptide is what we require, and for the proper folding we need some reconstruction and some finishing work. Rough endoplasmic reticulum has a single regulated system to prevent non-functional proteins being forwarded to the pathway for the secretion or to the other organelles. New proteins that cannot be folded or assembled properly by chaperones undergo ER-associated degradation. So this is an important line, that new proteins that cannot be folded or that cannot get assembled, they undergo a degradation process in which unsavable proteins are translocated back to cytoplasm, cytosol, conjugated to ubiquitin, and then degraded in by proteosomes. So even this is how the unusable protein, the useless proteins which could not complete its finishing work, they go to the cytoplasm. In cytoplasm we have an amino acid called ubiquitin, and it gets conjugated with the ubiquitin and goes further on degradation with the help of proteosomes. As mentioned, proteins synthesized in the rough endoplasmic reticulum can have several destinations. We studied about we just checked in the last paragraph about its destination going to the mitochondria or lysosomes, etc. Intracellular storage, example in lysosomes and specific granules of leukocytes. Provisional storage in the cytoplasmic vesicle prior to exocytosis, example in the pancreas and some endocrine cells, and as an integral membrane proteins. Diagram in figure 2.12 shows a few cell types with distinct difference in the destination of their major protein products and how these differences determine cells' histologic features. So let's quickly check figure 2.12. Oh, so here's figure 2.12. We have erythroblast, we have eosinophilic leukocyte, we have a plasma cell, we have pancreatic acinar cells. Okay, we are going to study all these cells in a separate chapters again. So let's wait a bit. Cytoplasm. Now we have something important is smooth endoplasmic reticulum. We'll study all the medical applications at the end of the chapter, and so that we have all the medical applications noted down together, all together separately. I hope that you're taking some kind of notes. Pause the video wherever you want and just take some notes. Right, so let's now get into rough endoplasmic reticulum. Rough endoplasmic reticulum, regions of that lack bound polyribosomes make up the smooth endoplasmic reticulum. Sorry, we are reading now the smooth endoplasmic reticulum, which is continuous with the rough endoplasmic reticulum but frequently less abundant. Lacking polyribosome, this is the most important. Lacking bound polyribosomes, that's what we need to remember about this. SER is not basophilic and is best seen with the them. Yes, so here is an important part. We need them here, the transition electron microscope, to see rough smooth, because we could see rough endoplasmic reticulums because the ribosomes on it stain yellow, stain basophilic, but we don't have polyribosomes, so smooth endoplasmic reticulum cannot be stained, unlike the cisternae of rough endoplasmic reticulum, as I said. SER cisternae are more tubular or sac-like, with interconnected channels of various shapes and size rather than stacks of flattened cisternae. So I think this part is important. In SER, we have sac-like cisternae, whereas in the rough endoplasmic reticulum, we have flattened cisternae. SER has three main functions, which vary in importance in different cell types. Enzymes in the SER perform synthesis of phospholipids and steroid, major constituent of the cell membrane. This is super important. We often ignore smooth endoplasmic reticulum because it's not playing the major role in the polypeptide production. But look here, we have enzymes that are in the smooth endoplasmic reticulum, which synthesize phospholipids and steroids, and we know how important these two are in a cell. Also, these lipids that then transferred from smooth endoplasmic reticulum to other membranes by lateral diffusion into adjacent membranes by phospholipid transfer proteins or by vesicles which detach from the SCR for movement along the cytoplasm and fusion with the other membranous organelles. In cells that secrete steroid hormone, example the adrenal cortex, SCR occupies a large portion of the cytoplasm. So any cells whose role is to produce enzymes, we see an abundance of SER here, the smooth endoplasmic reticulum, because of its major function in synthesizing phospholipids and the steroids. So, other smooth endoplasmic reticulum enzymes, including those of the cytochrome P450 family, allow detoxification of potentially harmful and exogenous molecules such as alcohol, barbiturates, and other drugs. In liver cells, these enzymes also process endogenous molecules such as component of bile. This is super important. Super important. The detoxification through the cytochrome P50. I'm sorry, to the cytochrome 250 P450 family molecule. SER vesicles are also responsible for the sequestration and control of release of calcium 2 plus, which is part of the rapid response of the cell to various stimuli. We need calcium to be released sometimes. We'll study in pathology why we require calcium, like sometimes acid in stomach requires calcium to balance it and cool it down, and many other pathologic requirements and stimulus are present where we need an urgent release of calcium ions, and the cells which are abundant in smooth endoplasmic reticulum has a capacity to produce calcium ions, which is part of rapid response to of cells to various stimuli. This function is particularly well developed in which striated muscles, where the SCR has an important role in contraction process and assumes a specialized form called the sarcoplasmic reticulum. We'll study this in chapter 10, but this is an example where we have abundance of the SCR. All right. So this was about the endoplasmic reticulum. This we studied that what endoplasmic reticulum is, what is the meaning of the word reticulum, how is it important in the process of producing polypeptide chains because of the presence of ribosomes on it. And when we have ribosomes on an endoplasmic reticulum, we call it the rough endoplasmic reticulum. And when we don't have ribosome on it, we call it smooth endoplasmic reticulum. This does not mean smooth endoplasmic reticulum is not of use. Its most important functions are three. First one is it has enzymes which produce what? Polypeptides and steroids. Also, its other important function is detoxification with the help of some enzymes. What enzymes of family P450 cytochrome P450. And its third important use is sequestration and release of calcium ion whenever required with the stimulus. All right. So next one is Golgi apparatus. Let's see what's the role of Golgi apparatus is. The dynamic organelle called Golgi apparatus or Golgi complex completes post-translational modification. We need to highlight this. Post-translational modification for proteins produced in the rough endoplasmic reticulum and then packages and addresses this protein to their proper destination. The word packages and addresses is also important, to their proper destinations. The organelle was named after histologist Camillo Golgi, who discovered it in 1898. The Golgi apparatus consists of many smooth membranous sacchi, some vesicular and others flattened, but all containing enzyme proteins being processed. In most cells, Golgi complex are located near the nucleus. So we have smooth membranous sacules, some vesicular and other flattened but containing enzyme and protein being processed. Oh, sorry. As shown in the figure 2.13, the Golgi apparatus has two distinct functional sides or faces forming formed by the complex traffic of vesicles within cells. Material moves from the rough endoplasmic cisternae to the Golgi apparatus in small membrane enclosed carrier called transport vesicles that are transported along cytoskeletal polymers by motor proteins. So here is a transport vesicle, the proteins, the material basically which is protein that comes from the rough endoplasmic reticulum cisternae to the Golgi apparatus. It forms in a small vesicle. We're going to call them membrane, which are transport vesicles, and these are membranous. So we'll just highlight what are transport vesicles. The transport vesicles merge with the Golgi receiving region, the cis phase. So the structure of the Golgi here is very important. Let's check figure 2.13 quickly. This is figure 2.13, So okay, so we have two phases here, and we have a cis phase here is visible, and we have a trans phase here visible. And cis phase is where the protein comes from the endoplasmic reticulums to the Golgi here. Okay, so always remember the cis phase is the receiving phase. Let's highlight the receiving region, the cis phase, or the on the opposite side of the Golgi network, it's shipping or trans phase. Trans phase of the Golgi is the shipping phase. Larger sacules or vacuole accumulate, condense, and generate other vesicles that carry completed proteins products to the organelles away from the Golgi. The vacuole accumulate, condense, and generate other vesicles that carry the completed protein products to the organelles. So these protein products go to the organelle in the form of vacuoles. Formed of formation of transport vesicles and secretory vesicles is driven by assembly of various coat protein, which also regulate vesicular traffic too through and beyond the Golgi apparatus. Okay, let's see what regulates this traffic. Forward movement of vesicles in the cis Golgi network of saccule is promoted by coat protein 2. We need to remember the role of these proteins. So forward movement is promoted by coat protein 2, while retrograde movement in that region involves the COP protein 1. Other membrane proteins important for directed vesicle fusion includes various RAP protein and other enzyme receptors and specific binding proteins and fusion promoting proteins that organize and shape membranes. So RAP proteins here are important in shaping membranes. Okay. So depending on the activity of the protein, all right, everything okay. Depending on the activity of the protein, vesicular vesicles are directed towards different Golgi regions and give rise to lysosome or secretory vesicle for exocytosis. So Golgi is what gives rise to lysosome or secretory vesicle for exocytosis. This is important. I'm sorry. As indicated in figure 214, Golgi saccules at the sequential location contain different enzymes at different cis, medial, and trans level. Enzymes of the Golgi apparatus are important for glycosylation, sulfation, phosphorylation, and limited proteolysis of protein. So we have to remember whenever we have the word silation means we are cutting it down, or we have lysis means we are cutting it down into. So here word glycosylation means breakdown of glucose. We have sulfation, phosphorylation, and proteolysis, basically breakdown of proteins. These are the works carried out by enzymes which are different on the different locations in the Golgi. So we have different proteins at the cis, at the medial, and the trans region. Along these activities, the Golgi apparatus initiates packing, concentration, and storage of secretory products. So packing, con- concentration of the secretory products, and enzymes of the Golgi are responsible for silations. Protein movement through the Golgi and control of protein processing are subjective of active research, or are subjects of active research. Oh, so the protein movement, if you are interested in any kind of a research work, you should consider this. Secretory granules. Secretory. Let's check off what are the secretory granules which are produced from the Golgi apparatus. The in the form of vacuoles or something like that. Let's take a look. Let me check if we have any question in our chat. All right. So what are we going to do is we will get done with the secretory granules, and the next one from lysosomes we'll have a new lecture again. So we'll wrap up after the secretory granules. All right, we are done with the Golgi apparatus. Let's get into secretory granules. Originating as condensing vesicles in the Golgi apparatus, secretory granules are found in the cells that store a product until it's released by exocytosis. It is signaled by a metabolic, hormonal, or neural message, which is a regulated secretion. So, secretory vesicle is basically, it's a form of storage of any particular product. This can be protein, and it is stored until it receives a signal for its production, for its requirement. So basically these are storage granules, secretory granules. The granules are surrounded by the membrane and contain a concentrated form of secreted product. So of course we need this product to be concentrated so that we store it in the less space. The contents of some secretory granules may be up to 200 times more concentrated than those in the cisternae of the rough endoplasmic reticulum. Well, rough endoplasmic reticulum, the proteins are barely concentrated. They are in fact just folded. But we have 200 times more, condensation when we come to secretory granules. All right. So this is a visual of a secretory granule that has been produced from the Golgi. So we have protein that produced in the rough endoplasmic reticulum. It moved packaged in a specific way where it's need to be stored before it's being exocytosed, and it is now here stored in the form of secretory granule with dense content of the digestive enzymes are also referred to as zymogen enzymes. So the secretory enzymes, basically the secretory material with the, oh sorry, the secretory granules with dense content of digestive enzymes are referred to as zymogen granules. We'll study about this in chapter where we study about digestion. What are zymogen granules? So the as I said, the contents in the secretory granules can be proteins, can be enzymes, can be hormones, and in the case of zymogen granules, this secretory granule consists of a digestive enzyme. All right, so let's wrap here. We are already around 40 minutes or something, and I don't want to stretch things to more than 40 minutes in one particular lecture. Next, we're going to study lysosomes. Uh, let's highlight the zymogen granules just in case it is a question, but we'll have it in detail in later chapter. We'll study next lysosome. Then what we study next is the mitochondria, the proteosomes. Then we have mitochondria, and then we have peroxisomes, and that's it, I think. Then we move on to the cytoskeleton. So three more important, four more actually important organelles are left, and we will take almost an hour more to finish all this important. We'll study the glycolytic pathway a little bit when we read about the mitochondria. So thanks for watching. See you on the next lecture.